century, Leeuwenhoek, a Dutch tradesman and lens maker, constructed simple yet powerful microscopes, each small enough to fit in the palm of a hand, but strong enough to reveal an entirely new world. Through his handcrafted lenses, he became the first person to observe the living microorganisms, which he called animalcules, in rainwater, pond water, and even in dental plague. His discoveries were transformative for science, as no one had ever seen such life forms before. Fascinatingly, Leeuwenhoek documented his findings with great details and sent his handwritten letters and microscopic observation to the Royal Society of London, one of the most prestigious scientific institutions of that time. His detailed reports, later published in the Philosophical Transactions of Royal Society, marked the birth of microbiology as a scientific discipline. So today, we celebrate International Microorganism Day. We also celebrate the human curiosity, innovation, and the endless desire to explore the unseen.
Now, I would like to call upon the Dean of the School of Health Science and Translational Research, Dr. Romit Sarkar, to the stage to say a few words. Please, sir.
Thank you, Dr. O'Neill. Thank you, Prantika. Respected Speaker of the Day, Dr. Ognivo Majumdar, my esteemed colleagues, and my dear students. A very good afternoon to all of you. It's really a pleasure and honor for me to be here to observe this International Microorganism Day. I understand that actually this International Microorganism Day is on September 17th, I guess. So due to several reasons, we couldn't accommodate the observation on that particular day. But I would request all the faculty members and HODs of Microbiology and Biotechnology Department to mark that in the calendar so that next year onwards we can observe the day on September 17th itself. Anyway, so personally, you see, I would say that microorganisms are very close to my heart. The reason is that whatever little research I have done so far in India and US is always hovered around microorganisms. Or rather, I should say that whatever little biology I understand, it's also mostly revolves around microorganism in different form, bacteria, virus, fungi, etc. So you see, microorganisms are the world of microorganisms, or as you say, the unseen world. It's a fascinating world and magnificent world that the students you maybe already know or you will get to know in subsequent years. It's the only life form, apart from human life form, that we always carry with us in the form of gut bacteria or in different other organs as normal flora. How do we get this normal flora? Say, for example, in first instances, mostly when the baby is born and passes through the vaginal canal, they accumulate those from the mother. And similarly, it's sad, but it's a reality that whoever is assembled here in this hall today, maybe after reaching to the mature age, most of us will die due to septicemia because of multi-drug resistant bacterial infection. So, microbes are there during the birth. Microbes are there at the time of death. Sometimes as friend or sometimes foe. And in between periods, they are everywhere. From the applications point of view, in food processing, in fuel production, in sustainable ecosystem, to maintain our physical and mental well-being, and also for different types of sufferings in terms of infectious diseases. There is a huge impact on agriculture, huge impact on livestock production. Everywhere they are there. So in a sense, that kind of controlling all our all our moves, all our, as I said, physical and mental well-being. Every passing day, we came to know different information or gathering about microorganism. Maybe 2 decades back, it was not clear how microorganism is controlling our psychology, our mood, etc. It's now pretty much evident that they are there due to this gut-brain axis. They're controlling. So maybe whatever the way you are, your character is kind of because what type of microorganisms you harbor. So I would say that this type of program, Observations of International Microorganism Day, provides us the opportunity as a student, as an educator, as a scientist to reflect our role, to understand microorganism, to harness microorganism, and above all, to respect the world of unseen, as he said. This also gives us the scope for opportunity to ask questions about microorganism, to explore different innovative ideas, how we can use them, how we can control them. So that is, I hope, from the discussion or lecture we'll hear from Dr. Majumdar, we'll be able to understand in a better way. I would like to thank Dr. Atri Ghosh and her colleagues from the Department of Microbiology and Biotechnology and all the students who has participated to make this program a huge success. Thank you. Thank you very much.
Thank you, sir. Now, to mark the beginning of this special day, let's start with a gesture that symbolizes life and growth, the watering of the plant. I would like to invite our esteemed guests and faculty members to come forward for the plant watering ceremony.
Many thanks to everyone present here. Now, let's move ahead to our first session. It is an immense pleasure to introduce our distinguished guest, Dr. Ognivo Majumdar. Dr. Majumdar serves as a medical microbiologist and infectious disease researcher at the National Institute for Research in Bacterial Infections, Kolkata, under the Indian Council of Medical Research. His work is focused on emerging and re-emerging pathogens, zoonotic infections, antimicrobial resistance, as well as the One Health approach. He has significant contribution in several multicentric studies and clinical trials, as well as national disease control strategies. He oversees diagnostic services, outbreak investigations, EQA programs, IVD kit validations, biosafety, biosecurity, as well as specialized trainings. Under the aegis of nationwide programs like NACO and the Virus Research and Diagnostic Laboratory Network, his contributions bridge laboratory science with real-world clinical and public health impact. Sir, we are truly honored to have you with us today. The lecture will be followed by an interactive session, and then we'll continue with our Rangoli competition and the photography event to celebrate creativity and science together. Sir, I warmly welcome you on stage to deliver your lecture. I would also like to call upon the head of the Department of Microbiology on the stage for felicitation. Thank you. So please.
Good afternoon all. So it's an immense pleasure to be here speaking to the undergraduate students after a long time. Usually most of the interactions are with the postgraduates, scientists and other faculties. So it's after a long time that I'll be talking with the undergraduates, the same place I have been once. And so I made my presentation in such a way that it will be a popular talk, an interactive one, if it permits, I think. And for any question, at any time you can stop me, or we can take the questions later also. So this topic, from petri dish to pandemic, biosafety and biosecurity, concerns in the 21st century. So I deliberately chose this topic because what I see when a new fresher joins our institute and comes to work in a lab, mostly the clinical microbiology labs, there is a gross disregard to the biosafety and the biosecurity protocols. It's not their fault, it's not built in our curriculum per se. Because when you are working with a non-pathogenic organism, now you move on to the next level, you pass round of interview or any admission to a new institute, and you go into working with a clinical pathogen or a pathogen with a potential to cause disease. The concerns are different. You have to follow the lab protocols, the biosafety and the biosecurity guidelines. Can anybody tell me? I think everybody knows this symbol. Everybody has seen somewhere, in movies, in games. So what is this symbol? Biohazard symbol, yes. So what does this mean? Anybody? What this symbol means? Any guess? It does not mean anything. I tell you why. This Charles L. Baldwin of Dow Chemicals and Robert S. Runkley of NIH in USA. There was a competition to create a symbol which is very striking and that will be instantly recognizable and will be remembered by the common people. So this was selected in a competition like thing at that time, so that this symbol became the symbol of biohazard, and this is such a symbol that once you see it, you'll remember, and it will tell you that this is something which you should avoid or you should be careful of. So what starts in a small petri dish in a lab can end up affecting millions of people around the world. So this picture of the world shows the newly emerging, the red ones, the re-emerging or resurging, and the deliberately emerging, the black ones. So what is emerging and re-emerging diseases? emerging are the new ones that come up, and the re-emerging are those diseases were in the past, but due to some different actions, man-made or natural, they are again coming up. In India, you can see typhoid fever is there, cholera is there, and the Nipah virus. I think every now and then we can, after 1 or 2 years, we can see that Nipah virus is being detected in Kerala. Earlier, Nipah was detected in West Bengal also. In Nadia district and in Shiliguri. And Nipah is very fatal. So ICMR, the team has located, there is a separate field office in Kerala who works on this outbreak, and that whole team mobilizes as soon as such an incident occurs. But still it is elusive, and we can only control that once it has happened. But to predict that and to prevent that is very difficult. So you see these organisms, many of the organisms here, they are well-established pathogen. People work with them around the world, but they can pose a grave danger to the people, and they have the potential to cause pandemic even. So the promise and the peril of microbiology, it's a double-edged sword. The same microbe that heals can also harm. It depends on how we handle it. It's up to us, the scientists, the researchers, or the doctors, the clinicians, how we are handling these pathogens so that they come to our benefit and not to harm. So this timeline, you see, it's a very generalized timeline of how the microbiology has proceeded over the last few centuries. As in our introductory opening talk, it was told that Robert Leeuwenhoek was the first to show us what's the real world of microbes are. But it took almost 2 centuries to really acknowledge how they are affecting our daily lives. The early microbiology, that is understanding of the germs, mainly began in the 19th and the early 20th century. And after that, there was an exponential increase of the research, knowledge, and all the works that we are doing now in the field of microbiology and biotechnology. Then came the vaccine or the antibiotic revolution. It started with penicillin, and afterwards, one after another, there was a booming discovery of antimicrobials. And as soon as the antimicrobials are discovered, they came with the resistance. But that was controlled use of microbes. And then came the recombinant DNA era, where the engineering of the microbes began. And then it went one step ahead. We became gods. So the dual-use technology explosion, where engineering the traits and which has potential weaponizing capability began. And now we can see every few years there is a outbreak, pandemic, and this is increasing. So coming to biosafety and biosecurity, what is biosafety? The definition stands as the application of safety principles to a laboratory in which biological organisms or materials are manipulated to protect the workers and the environment. So there are the workers and the environment that we need to protect. So its focus is on the safety. The safety aspect is the main focus here. And what are the 3 pillars? To ensure that the lab we are working is safe. First is the good laboratory practice and techniques, like the standard operating procedures, the induction and the training. This is very vital. One who is coming into a lab to work first, they have to be trained first. And induction training is must in a lab where a potential dangerous pathogens are handled. Then comes the safety equipment, like the biological safety cabinets, the personal protective equipment, PPE, and the facility design and construction where appropriate workflow and controlled airflow is mandatory. This has to be ensured. These are the three pillars on which a biosafety stands. And there is another word, the biosecurity. So the discipline addressing the security of microbial agents and toxins to reduce or eliminate the threats posed to human or animal health, environment, and economy by deliberate misuse or release. See the word deliberate. So to prevent that, the focus is on the security. And these are some of the key aspects that we need to ensure in a lab to ensure that biosecurity is there. Like the physical security, personal security, one who is entering and leaving the lab, the material control and accountability, the stock you are maintaining, the transport security, and information security, like the IT, which is maintaining the catalog or the total inventory. And these all comes under the program management. So these are the two, biosafety and biosecurity by definition, and the key aspects of that. So this, I love movies, Hollywood movies. I just want to play this. Can I play from here? I don't know if it is played from here. Yeah, please. Sound is missing, I think. So this is one of the trailer of a incident which happened actually in US. It's a web series developed on that.
Settle. They lie in wait. What we're talking about is a ticking time bomb. I saw it with my own eyes. Get out.
America has many enemies. I'm here to warn you. An attack is inevitable.
I need a team at Capitol Hill right now.
FBI, stand and calmly exit the building.
We've got a full-blown bioweapons attack on our hands.
I need you to catch who did this.
Even in a vacuum, how do you trust someone with the safety of our nation?
I think you're letting all this get to you.
I just think something's amiss.
I have to find who did this.
There are ways to protect ourselves.
I encourage everyone not to overreact here.
How bad is this?
It's 100% lethal. We can't afford to waste time.
What if the anthrax killer's one of us?
What is the incident? Anybody? It's based on true event. When did it happen? Anyone? It's 2001, just 4 weeks after the 9/11 incident. Anthrax spores were sent by mail to the US senators and 4 media houses. Around 5 people died due to the exposure, inadvertent exposure, and 17 were fell ill and afterwards recovered. And if you go to the history of that, the final verdict was it started in 2001. The FBI took over the investigation with so many resources, and in 2010, probably, it came out that the most prominent scientist working on anthrax is the culprit. And irony is that that person was the first one to call to be in the part of the investigation. He was found to be schizophrenic, and that was the case. Next slide. Next slide. So there are several such incidents of biosecurity and biosafety breach that happened. Some of the notable interesting ones are this 1981 accidental release of anthrax in USSR. That was a facility under the Soviet regime, and there was an accidental release and many people in the neighboring area died. In 1989, lab workers exposed to the resistant Ebola virus. It's a close cousin of Ebola, but it's not so much lethal. Ebola, I think everybody knows that. And so it had the potential to cause a huge damage, but luckily they escaped. And it was obtained from monkeys from other countries, imported monkeys. 2001, this is called the Amerithrax, the clip that you just saw. Afterward, in 2014, CDC anthrax exposure, after live samples were sent to the lower level laboratories. This happened in CDC Atlanta itself. 2011 to 2012, there were controversy regarding the H5N1. It's an avian influenza. It's usually not transmitted to the human. In India, there is no such cases. It's very less common. But it can be, this is an avian influenza. It occurs in birds. But if any gain of function, it can be transmittable, gains transmissibility in the mammals or humans, it can cause havoc, like another pandemic like COVID. So there were some publications regarding this, that some engineering work has been done, and this gained criticism after that. 2019, there was accidental brucella leak from vaccine plant, Lanzhou, China. And 2022, attempted purchase of pathogens through dark web, through cryptocurrency. This is now the trend. It's happening. Now tell me which one is biosafety and which one is biosecurity risk. The first one, accidental release. Biosafety or biosecurity? Safety. It's safety. Accidental release. Then, lab workers exposed to the resistant virus. Yes. Then, 2001, anthrax male episode. Biosecurity. It's a deliberate attempt. Then after that, CDC anthrax exposure. It was sent to a lower level lab from a higher level lab. Safety, it was not a deliberate one. They were working in the same complex. And after that, H5N1, they were engineering that to make it more lethal. Security, exactly. And accidental brucella leak. And the last one. So the concept is clear. What is biosafety and what is biosecurity? So why it matters more than ever? We are living in the 21st century and we are more concerned with these biosafety and biosecurity aspects. Because there is a rapid pathogen research. The dual use research concern, that is synthetic biology and the gain of function studies. This CRISPR technology that we will come across, it causes insertion of genes into a microorganism, and that can result in gain of control or gain of function. Okay, so gene therapy, targeted gene therapy, that's a huge medical aspect is there, but it can also can cause grave danger. There is global travel, so rapidly for any infection to spread across the world, it hardly takes a few hours. A flight from here to US through Dubai, and three continents are already affected. And there are lessons in the past, like the SARS. It broke out in Hong Kong, and it's the earlier one, not the SARS-CoV-2. It's the SARS first one. Then came the MERS. MERS is still there. The Ebola, you can hear Ebola after a few years, every year, after some gap in Africa. And COVID-19, you all know. And also, currently, there is a danger of cyber biosecurity and synthetic biology concerns, like there is a hacking attempt for the genomic databases and the pathogen repositories in the cloud, getting the genome from there, editing them, and then procuring them. Like unauthorized orders of synthetic DNA resembling the restricted sequences, like the smallpox, which has already been eradicated from the world. Now what happens if a more lethal version of the smallpox or the same virus comes? There is only 2 places where smallpox repository is there. And all other places it has been destroyed. One in CDC Atlanta, another in Russia. These are highly secure facilities in these 2 places. And why should you care? Because some of you are already working and the rest will work with pathogens someday. Because understanding the risk help you design safer experiments. And also, microbes don't respect borders. See, the local lab accidents won't stay local. It will spread. The COVID-19 pandemic has shown every one of us that we have to be concerned with these things. This bat picture, can you tell why bat is so important in biology, in microbiology as a whole? Bat should be a symbol of microbiology. Because bat is a unique mammal or an animal which can harbor many different viruses, lethal viruses, that does not cause any harm to that animal. But it can transmit. Even it acts as an incubator where genetic reassortments can also occur. Like influenza. From pig is the incubator for the genetic reassortment of the influenza viruses, different strains. Those are carried by the birds. So this Ebola, Nipah, SARS-CoV, many of these have their origin from bat. I'll come to a different movie later on and I'll tell you. So in a clinical microbiology lab, or a clinical lab, any lab, it's not only microbiology, this is the flow of samples. The red one, the dark red one, you can see it's a pre-analytical phase where from the patient the sample is collected, then initial processing is done, storage, packaging, transport to the lab. The blue one is the lab where final processing and testing is done, and the black one is the storage and the discard, which is the post-analytical part. Can you tell me in this process where the biosafety lies? From starting to end, where the biosafety lies? And where does the biosecurity lies? This is how every sample is processed. You go to a clinic, to a laboratory for any investigation. This is the flow of samples, more or less. So I'll tell you. So this is the realm of the biosafety and the biosecurity. It begins from the patient assessment and runs till the discard. So it's no place that you can be less concerned about biosafety and biosecurity. And there comes the guidelines. The WHO is there, the CDC, and the Indian DBT guidelines. And for the COVID also, they are updated, and we have to follow the guidelines for this biosafety and biosecurity. And the ministries is also concerned about this. The Ministry of Environment, Forest and Climate Change, the Biomedical Waste Management, this falls under this. The Pollution Control Board, they ensures that we follow those guidelines. The DBT, under the Ministry of Science and Technology, they have these bodies. The Recombinant DNA Advisory Committee, the Institutional Biosafety Committee. This is very essential for any research institute or any institute working with pathogens. The Review Committee on Genetic Manipulation for any biotechnology or genetically altering of any organism. You have to have this clearance. The Genetic Engineering Appraisal Committee, the State Biotechnology Coordination Committee, and District Level Committee. And also the ICMR under Department of Health Research has come up with the guidelines for the high containment labs. Like these guidelines, they have brought out with DBT DST for the BSL 3 level labs. So these bodies ensure that you have to follow these guidelines. Another part, apart from the flow of sample, also there one part is also included there is the sample packaging and transport. So why it is important? So some lab is sending a sample to another lab, referral lab. Suppose in our case, when there was a Nipah scare in West Bengal, so one of the hospitals in Kolkata sent suspected Nipah positive sample, suspected one, because only from the clinical features they could make out that it may be Nipah positive one. So that sample came to us. Or forget about Nipah, it's in the early days of COVID-19 when nobody knew what level of virus or what potential this virus has. That sample was reached in our institute. So initial handling, initial screening, we have to send that to NIV Pune, National Institute of Virology Pune, the apex body for the viruses. So there we have to transport. But who is transporting? The Sequel, Blue Dart, these are the courier company. And who are working there? Common people. So we have to ensure that the samples are transported in such a way the middle person should not be in harm. So this packaging supplier or the operator and the carrier, that comes in between the consignor and the consignee. So there are strict international guidelines that we have to follow for this sending of the transport. And also WHO has come up with this, and this is regularly updated. Every 2 years, they come up with a new version for the guidance on regulators for the transport of infectious substance. If you try to carry one vial of any pathogen or any microbe with you, can you pass the security check of airport? No, you cannot. Either it will be smuggling or you will be detected, because you do not have the proper clearance and also don't have the proper way of carrying those organisms. So there are strict guidelines that is governed throughout the world. These are the guidelines for the airway. Most of these dangers are associated with the airway transport. And also there are separate guidelines for seaway and landway also. So there are United Nations guidelines from Civil Aviation Organization, the IATA, the International Air Transport Association, and also India have the DGCA guidelines that are governing all these things. And during the COVID-19, there were some revisions and of this. So we have to know even for us transporting a sample from one place to other. We cannot just send the sample or carry that sample from there. So what we are talking about, samples. So there are other things also which come under this purview. These are called dangerous goods. These are the international symbols associated with these dangerous goods, and also the coding. This code is universal. And we are mainly concerned with these two things. One is the infectious substance, which falls under category 6. And also the miscellaneous dangerous goods, because sometimes we send samples or any microbe in a cold chain. That may be ice packs, that may be dry ice, and also liquid nitrogen. These dry ice and liquid nitrogen are dangerous goods. So you have to again follow. Even if you are sending a non-pathogenic organism, but it's under dry ice or liquid nitrogen, you have to follow the same guidelines. Now, coming to the risk groups and the biosafety levels. So these guidelines have divided all these organisms into 4 risk groups. The risk group 1, agents not associated with disease in healthy adult humans. These are the agents which we usually deal from a medical college point of view, I can tell in the demonstration classes. In the practical demonstration classes, we deal with the risk group 1 pathogens. I still remember in my first day of practical, I was given curd, dahi. Why? Microbiology practical, curd was given to see the cells, lactobacillus cells under the microscope. So these are the agents which are not pathogenic, and these are handled in a risk group 1 facility, like our demonstration classes. Then comes the risk group 2. These are the clinical pathogens which can make us ill. But there are medicines or treatments or vaccines available for them. So this is usually the most of the clinical microbiology labs or bacteriology labs, they work with this type of pathogens. Then comes the risk group 3 pathogens. These are serious or lethal to human health, and which preventive or therapeutic interventions may be available. And the risk group 4 is the highest level pathogen, where no such interventions is there. So anthrax falls in which risk group? Anthrax, it's in 3. And Ebola, Nipah, these falls under 4. Okay. Bacteria do not fall in the risk group 4. Those are the domains of only the exclusive to the viruses. And coming to this, the biosafety levels come. So as per the risk groups associated, there are 4 biosafety levels. But remember one thing, this risk group does not always equate to the biosafety levels. Why is it so? One pathogen which is endemic in a region, like it's very common in a region, when it goes into another country, another part of the world, the risk gets elevated. We have to make them fall into higher level of biosecurity. Suppose Klebsiella pneumoniae is one of the pathogenic organism. Many of you have heard, I think. Or suppose Escherichia coli. This is something everybody has heard. So one you are using for your daily experiments, for cloning activities. One which is making you ill, you are taking a medicine and getting relieved. And one E. coli which has gathered so many antibiotic resistance genes in them that it is multi-drug resistant. Same organism can fall into 3 different risk groups, and so different biosafety levels need to be used to handle the same Escherichia coli. So these divisions has been formulated based on this pathogenicity of the organism, mode of transmission, local availability of treatment, and local availability of effective treatment and preventive measures. So I come to the movies again. So there is one clip in one movie, it's called Outbreak. Though it's not entirely what the levels are, but you can have an idea of the 4 levels at a glance. Dada, play করুন. So these are not entirely correct, but one thing you can appreciate, that as the level increases, how the protection, the security, and the equipment changes. That is, you can appreciate these things. So this is true exactly. Next slide. As the level increases, we have to ensure to maintain these biosecurity levels, our practice, our dresses, our equipment, all are compatible with this. What is slide? What is slide? What is slide? So we have a BSL-3. Yes, we have BSL-2, 1, and we have the highest BSL-3 lab in our institute, which is operational. It's a modular one. This is how it looks from outside. And the laboratory here, if you go into the laboratory, you have to pass through 2 ways of changes of air. So there is a 2-door, interlocking door, which you have to pass before you can enter. And even when you exit, you have to pass through 2 doors, then you can exit. And you have to take a shower also while exiting, so that you don't carry any level 3 pathogen outside. So there is one clip where I want to show you the things like transmissibility, like why a pathogen is more lethal than an other pathogen. So let's hear from Kate Winslet herself.
Now, we call that number the R naught. R stands for the reproductive rate of the virus.
Any ideas what that might be for this?
How fast it multiplies depends on a variety of factors. The incubation period, how long a person is contagious. Sometimes people can be contagious without even having symptoms. We need to know that too. And we need to know how big the population of people susceptible to the virus might be.
So far that appears to be everyone with hands, a mouth, and a nose.
Once we know the R naught, we'll be able to get a handle on the scale of the epidemic.
So it's an epidemic now. An epidemic of what?
We send samples to the CDC.
In 72 hours, we'll know what it is. If we're lucky.
Clearly, we're not lucky.
What do we got?
A young woman in Minnesota recently traveled to China. Son also died. As of this morning, 87 cases, 15 deaths.
Did you have a good Thanksgiving?
Yeah, good.
You?
So this suit they are wearing, it's a positive pressure suit. So what's the uniqueness in them? The pressure, there is a pump which forces pressure inside. So the inside is in a positive pressure. The environment or the lab is in negative pressure. So that there is no chance of anything from outside getting into that suit. Even you can see some pipes coming down from the roof. What are those? Those are their oxygen supplies. They are entirely enclosed. If they have to move from here to there, they have to change their pipe from here and then go to that place and then again attach to that. It's very difficult working in a BSL-4 lab. We have one BSL-4 lab under ICMR at NIV, and the person who gets into that lab, they have to be medically fit, they have to take optimum sleep, optimum water before they enter the lab. And the number that she was talking about, the R naught, is a reproductive number for the pathogens. Like from one person, if it transmits to 3%. So this R naught becomes 3. And this depends on the resistance of the person to acquire the disease, the infectivity, the transmission potential, and more this number, the quicker this virus or the pathogen will spread from one person to other. What is slide?
Leaving you.
We have 47 cases and 8 deaths as of 5 this afternoon. It's a weekend. These numbers might be low.
So, but what you can appreciate one thing is that this is very costly thing. The more you go up, the cost increases. So how a developing nation or a poor country can afford such type of higher biosafety levels? So the WHO has come up with risk-based assessment, that you have to understand what is the risk of the pathogen or the work you are doing in your context. So here you have to know what is the consequence of exposure or the release of the pathogen, like severe, moderate or negligible, and what is the likelihood of exposure or the release, like unlikely, possible or likely. And you see the diagram here on the right side, a pathogen which is very highly likely of getting exposed or released with a very severe consequence that has to be handled in a higher containment lab. In the bottom diagram you can see the maximum containment measures are those which have a higher likelihood of release. So this is how you grade your work, what you are doing with the pathogen and the work, and this is how the containment measures are now being placed. So there are other things like laboratory design, biological safety cabinets, PPE, decontamination of waste management, the biosafety program that you need to address to maintain the adequate biosafety and biosecurity levels in your lab. So facility design, this is use of covings, like those ends that has to be rounded. No fabric surface in furniture. These type of chairs are not to be used inside the lab because any spillage occurs, it seeps inside, it stays there. You cannot decontaminate them. No curtains or blinds with absorbent surfaces inside the lab. Doors should be with vision panel and fire retardant, no wooden doors. Hand washing facility should be close to the exit so that you can hand wash and go out. The AC must ensure a comfortable working environment because you know you are wearing a PPE, lab gowns, and the temperature is, what it was, 31 or 35. It's very tough. Also, there should be proper labels and signs like this type of biohazard label that has to be affixed outside the door, and this is mandatory for biosafety level 2, 3, 4 level labs. And this kind of signages should be posted. These are international signage symbols that should be mentioned on every places where appropriate, like the prohibition symbols, warning symbols, mandatory symbols, fire equipment and fire action, and the safe condition and first aid. Same for this laboratory hazard signs. These are also international symbols that has to be adequately pasted wherever you are working. And the biosafety cabinet, you should be ensured that only having a biosafety cabinet does not guarantee protection. The person who is working in the biosafety cabinet has to know how to work in the biosafety cabinet. You have to know how the airflow occurs in a biosafety cabinet, the laminar flow. Working at the proper sash level. This glass has to be at the proper level. Never cover the air gills because from there the airflow occurs. If you cover them or put some object over the grills, then it will obstruct the airflow. This laminar flow won't occur. Minimize the rapid movement laterally. You can only put your hands inside and out. And also reduce splatter as much as possible. You have to know the work area. It must be in the middle. And the left should be clean and the right should be dirty. This work should be from the clean to the dirty area, not the reverse way. Also working with tubes and bottles and petri dishes, it should be in a slant condition and also with a cover, not in a vertical way. And also there should be burners or alcohol lamps must not be used there. And you can use this heater where you can, it's called a loop sterilizer. It's work on electricity. And also adequate PPE. This is the kind of PPE it used to be worn during the plague. in the Europe. The plague doctors used to wear that kind of PPE. Now it has been modernized. But do you know what kind of PPE to wear in which cases? These 4 pictures are of different levels. So this is based on the risk assessment. You have to know what type of condition you are handling, and like that you have to wear or choose the adequate PPE. So what is the problem in here? This picture, right one. Anybody can tell? What's the problem in here? Is there any problem? The person who is working there. Any breach in biosafety? Tell, you can tell. Yeah, they are working in a table. This is open. This burner is not also on, and there is also a gap between the apron and the gloves. The place is cluttered. So there are many problems in here, one who is working. So this is actually incident happened. Somebody took this very seriously, this PPE thing. This person came to give sample during COVID-19 pandemic in initial days. He couldn't get one PPE, but he has to wear a PPE. So these are biohazard bags, and I don't know what other things that is stitched together to come. This is the actual picture in our institute. This person came to give the samples. Coming to the healthcare waste, you should know that biomedical waste is 15% of healthcare waste that is generated in a hospital setting. These are as per the guidelines. You have the color codings, and you have to follow that strictly. Okay. So what is the most important step in biomedical waste management? These are the 4 steps: segregation, collection, pre-treatment, and treatment and disposal. What is the most important step? Treatment and disposal. What is the most important one? Most important is 1. No, segregation is the most important. If you don't segregate at the source, everything will go haywire. The other person should not segregate them. Okay. So what's the problem in the left one? Not? Okay, but do we need to be enclosed? Okay, cover is good. But the major problem, what is that? In the left one. There is no segregation, plastic, yes. And another thing is very obvious, that it should not be so much full that it will just spill over. Maximum 3/4. Then you have to change. And in the right one, what is the problem? You should not drag that bag on the floor. You have to carry that. So, and coming to the emergencies in laboratories, there are certain emergencies that you should be knowing. This may be natural or man-made. And if you do not act on emergencies within 3 minutes, you are not likely to act at all. So this is the right way to have a biohazard spill kit if there is any emergency or the spillage in the lab. This left bottom one should not be there. This switchboard protruded out, not be there. There should be an eye wash station. There will be shower outside the lab. That should not be covered with other materials. There should be a first aid box in the lab. And there should be a biosafety officer in each institute if there is any fingerprint injury or realistic injury. And also fire, you have to know what type of fire and where are the fire extinguishers are kept, and you have to follow the way to use a fire. A fire drill should be done periodically. And when not to fight a fire, you don't have a proper extinguisher, fire has spread beyond its point of origin, and your instinct tells you to get out. So these are the fire control systems that are usually placed in the lab, and it should not be obstructed. And this is a information system to alert, and it is kept in a wrong way. This is a not used fire tender system. There should be an evacuation diagram. You should map your facility for that. And the spaces, that is the fire exit, should be empty. And there should be glow sign which marks you to the exits. And all the equipment should periodically go safety checks so that whatever you are using, like the centrifuges, compressed gas cylinders, they are in a proper condition. And there should be an emergency action plan in your institute where everybody should know those action plans. And of course, building a culture of biosafety inside the institute, like regular training and refresher courses, clear SOPs and reporting culture, no blame, just learning, visible leadership commitments, and include biosafety in student curriculum. And also, Niti Aayog has came up with this document where it has been emphasized for these kind of biosafety trainings in future pandemic preparedness and emergency response. So what is the future? There is a One Health concern where human, animal, and environment interact and posing new challenges day by day. Also, the use of artificial intelligence that guided designs for tailored pathogens, and this could accelerate the dual use misuse. There are also community labs and do-it-yourself biological labs that should be made aware and should be put under control. And like we have a ransomware security for our IT systems, pathogen ransomware is also a newer concept that is emerging, where a cyber attack may occur and they can extort to give you some lethal pathogens. So finally, how to handle risk in labs, as to take the key away points. The PPE, it should be adequate and proper as per the risk level. The workplace practice should be proper, like entry and exit process, laboratory decontamination, emergency procedures, handling of the biomedical wastes. The administrative controls like regular immunization, security clearance of the personnel going inside the lab and going out. The medical surveillance programs, training and compliance to SOPs. And of course, the engineering of the labs like the directional airflow, the HEPA filtration, entry and exit, access point controls. And all these play equally in protecting the workers from an occupational exposure. So this is an incident which happened a few years ago. And see the report. It has come to the light that an official of Southwestern Railway had warned about serious flaws in the system 3 months ago, but nobody looked into that. This caused into such an accident and loss of lives. And this is not unique. There are other incidents throughout Indian labs, like from IISc, DRDO lab in Pune, then in Delhi and other labs. So we should work in such a way that this should be avoided. And finally, this is the takeaway message: the best designed and most well-engineered laboratory is only as good as its least competent worker. So we have to make this aware all the person who is working in the lab. Thank you. Anybody have any questions? I can take.
Thank you, sir. I hope everybody, they have uploaded their photographs in the given link, the photography participants. So now the pictures, it will be displayed on the screen one by one, and each student, they will have to give a 1-minute speech on their photograph. So
Hello.
everybody, can you please maintain silence?
The stage is open for the interactive session. If anyone from the audience has any question to the esteemed guests, they can ask. If anyone from the audience have any question to the esteemed guest, they can ask now. The stage is open for the interactive session. If anyone has any question to the esteemed audience, esteemed guest, they can ask now. Thank you.
Ritik Shadukar. I'm telling you the sequence. Ritik, Aditya Narayan Paul, Niladri Mondol, Deepa Basu Majumdar, Anushka Mehra, Avinav Chaudhuri, Shaunav Vishwash, Prathamesh Paul. I request all the participants to come near the stage. Ritik, Aditya, Niladri Mondol, Deepa Basu Majumdar, Anushka Mehra, Avinav Chaudhuri, Shaunav Vishwash and Prathamesh Paul.
Good afternoon. My name is Ritvik Shadukhan and I am from MBBT department, SEM 3, Biotechnology. So this is the microscopic view of a fungus which I actually took from my garden today morning only. So under the tree branch there is a fungus grown, so I just collected the sample, and this is the view of 40x view of the fungus, and we can see several microscopic spores and the hyphae and the network like hyphae which is surrounding the spores. So maybe this is in this mushroom species. I am assuming, actually I don't know this family. So it is the fungus view of 40x view of my fungus. Thank you.
Next, Aditya Narayan Paul.
Your time starts now.
Good afternoon, everyone. I am Aditya Narayan Pal from UG1 MB. I am here to present my photo, which I have took from my garden, which is based on chlorella, which is a unicellular algae. Chlorella is like a green hero. Chlorella is a chlorophyll microbe, which is from the algae family. It supports oxygen, and it removes carbon dioxide from the weather, and it also can synthesize its own food. And chlorella is a superhero, like for the space travelers, like it is used as a supplement for the protein and for the space travelers. Thank you, everyone.
Next, Niladri Mondol.
Hello. হ্যালো, আমি নীলাদ্রি মণ্ডল, আমি ইউজি 5-এ, আমি মাইক্রোবায়োলজি স্টুডেন্ট। তো স্যার এখানে এটা আমি 100x জুম করেছি, মানে 100x মানে ম্যাগনিফিকেশন করেছি। এটা একটা স্পাইরোগাইরা, আর এটা চারিদিকে ফরমিডিয়াম করেছে। অ্যাকচুয়ালি এটা মিক্স কালচার হয়ে গেছে, মানে এটা নিয়ে আমি ছবি তুলেছি বাস।
Next, Deepa Basu Majumdar.
Your time starts now.
Good afternoon, respected sir and ma'am. I am Deepa Basu Majumdar, UG 3BT. This image basically shows a microscopic view of a fungus obtained from a contaminated flask, and I observed it under 40x magnification. Thank you, sir, ma'am.
Next, Anushka Mehra.
Hello.
Your time starts now.
Greetings of the day to everyone present here. I am Anushka Mehra from UG3 Biotechnology Section A. The picture shown here is a magnification at 40x of a crystal-shaped fungi, which was taken from a contaminated flask. Yes, thank you.
Next, Abhinava Chaudhary.
Good afternoon, everybody. My name is Abhinava Chaudhary from UG5MB. The picture I took here is of lactobacillus at 40x magnification. The blue color we have seen over here is because of the crystal violet, which has been retained because of the thick peptidoglycan layer containing in it. And the reason to choose this microorganism as my photography competition picture is because my lactobacillus is beneficial for humans in many ways, like preventing gut bacteria and also improving digestions in humans, and also in females it helps in vaginal protections, and it also helps in curds. It also for using fermentation. Thank you.
Shona Bishash.
Hello.
Your time starts now.
Good afternoon, everyone present here. I am Sonav Biswas from UG3BT, and this picture here is the 100x magnification of Acidicia coli, a rod-shaped bacillus that is found in our guts. Many people here think that bacteria are actually bad, but some of them are actually good. This bacteria helps in breaking down foods that we eat. It also helps in the production of vitamins like K and B2, and it also helps in the protection from harmful bacteria. So my reason for choosing this picture is because it actually is our unseen friend that helps us from everyday life. Thank you.
Prathamesh Paul.
Your time starts now.
Good afternoon to everyone present over here. Myself, Prathamesh Pal, student of BSc Biotechnology, UG1, SNU. For this photography competition, I chose my microorganism as algae. In this picture, you can see I have shown a microscopic view of Euglena species in 40x magnification. Euglena is a genus of single-celled flagellated eukaryote that possess both animal-like and plant-like characteristics. The color of the organism is green due to the presence of chloroplast in them, which is used for photosynthesis similar to plants. The shape of the organism is like an elongated teardrop. The Euglena is commonly found in brackish water and freshwater environment that are rich in organic matter. I chose euglena because euglena are generally considered. Thank you.
Hello? The QR code for getting the certificate is going to be screened in the LED screen as well as the TV. You are requested to scan the QR code and download the certificate, which will be provided in your registered email ID. The QR code for getting the certificate is being screened in the LED screen as well as the TV screen. You are requested to scan the QR code as well as getting the
Volunteers, please maintain the chaos.
Hello? The Rangoli competition has already started from 2 p.m. I would like to call upon the esteemed judge to for the judgment of the Rangoli. I would also like to request all the students to visit the free space beside the seminar hall for seeing the Rangoli competition as well. Thank you.
বিকজ আমাদের টাইমিং খুব খারাপ, যখনই এসেছি তখনই জাজ এসছে, জাজের পেছনে এই 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 রেজাল্টের লিস্টটা হ্যান্ড ওভার এরপরেই তো রেজাল্ট।
Sports have been. আজকে বলে দেবে? হ্যাঁ।
ম্যাম ম্যাম একটা গ্রুপেতে 3 জন আছে, মেয়েটা কিভাবে দেওয়া হবে? ওখান থেকে মানে ইয়ে আসবে, মেডেলটা ওদের সবার, মেডেলটা কয়েকটা তুমি কোথায় বলতো? এই তো এই তো এইটা মেডেল কটা আছে বলতো? 10টা বলেছিলাম আমি।
Excuse me, everyone. Did you receive your certificates? In your mail ID, emails. And also we request you to stay for another 10 to 15 minutes. The prize distribution will be done, like for the photography competition and the Rangoli competition. If you all want, you can stay.
Hello. Hello, hello. Hello, hello. কাউকে একটা করে ডাক। Hello. Everybody, the previous QR code, it had some error, so everybody scan this QR code to...
Everybody scan this given QR code for getting the certificate. Everybody is requested to scan the QR code to get the certificates.
I would like to request Dr. Majumdar. So now comes the most awaited moment of today's event, the result announcement for our photography and Rangoli competition. I would like to call upon Dr. Ogniba Majumdar sir on the stage to distribute the prize. The winners for the photography competition. The third position, it goes to Niladri Mondol. The second position goes to Prathamesh Paul. And the winner, the first prize goes to Aditya Narayan Paul.
The winners for the Rangoli competition are going to be announced. So, the winners for the Rangoli competition are going to be announced now. The medals will be distributed later on. So the third prize for the Rangoli competition goes to Group 6. The winners are Swadha Chatterjee, Swarnali Dotto, Srija Sarkar and Niladri Mondol. The second prize for the Rangoli competition goes to Group 3. The prize goes to Poulomi Dash, Sampurna Borua, Devanjali Kundu, and Ghotok. And the first prize for the Rangoli competition goes to Group 1. The winners are Sweta Singh, Audrija Pramanik, and Regina De. Thank you. Hello? I would also like to call upon the winners of the photography competition to come up on the stage.
Assistant Director, Department of
Hello? I'd request the participants to settle down. Thank you. I would like to call upon the Assistant Director of the Departments of Microbiology and Biotechnology, Dr. Rajat Pal, for the keynote speech. Thank you.
Yes. A small correction, Shopnunil. It's not keynote speech. That's a very big thing. And I cannot give that. Okay, so what I can give is a vote of thanks. So a very big thanks to Professor Ognavo, sorry. I'm very sorry for spending so much of time for us and coming here, spending for almost all the day and judging the competitions, giving so insightful and knowledgeful lecture. I hope my students are encouraged. And a very big thanks for our Dr. Atrey Ghosh, the convener of all this event, total a day full event. She has made it possible. And the HODs, they have taken this initiative, and all my students, always whenever I am coming to dais for giving this vote of thanks, always I say that I should not thank my students. I should appreciate their hard work. So again, I'm appreciating all my students' hard work, and congratulations to the winner of these competitions. So again, in the next year, we shall, as our dean told, that we will keep in mind that in the next year we shall organize this International Microorganism Day in actually the day when it should be celebrated in 17 September, I guess. So let's see on the next year's 2026 Microorganisms Day. Till now, have a nice year. Goodbye.
All the students come up here near the stage for the photograph. All the students, please come up here.
इधर का हो जा भाई नहीं इधर हो जा लाइटिंग लाइटिंग हरी तरफ से आदि आदि को इधर रख अब दूसरा ट्रीट देना पड़ेगा ये लंबू साइड में चला जाओ हाँ आजा आजा हम अलग से दिखेंगे भाई अलग से नहीं दिखा दीजिए इतना बड़ा फ्रेम थोड़ी ना होगा आई